Hello, my name is Luis Quintana Murce. I have a lab here in the Pasteur Institute where our work is focused on human population genetics. So today I'm going to explain you the state of the art in population genetics related to humans, and we are going to see how the study of human genome diversity at the population level is very useful for various things: to reconstruct our history, the evolution of modern humans, to understand how we have adapted to different environments with a focus on infectious disease, to finally end talking about perspectives in precision medicine. So the last 15 years or so in human genetics have been characterized by major steps. We have the sequencing of the human genome, which was very important to localize genes, understand exon intron structure, but in no way gave us information about what we are interested, that is, diversity. Diversity between individuals and diversity between populations. That's why there has been a number of consortia that since they have been focused on understanding how human populations are diverse, from Africa to Europe to Asia and the Americas. And examples of that are the HapMap Consortium or the 1000 Genomes, whose main goals were to provide a genetic map of haplotypes in different human populations in order to be used for population studies on the one side, and on the other side for more medically applied issues. So what have we learned about these levels of genome diversity? We know today that each of us differs from the so-called human reference sequence by at least 10,000 mutations that change the amino acid sequence. And each of us can carry even 100 mutations that have been already associated with a genetic disease, fortunately at the heterozygote state. That's why, in most cases, we are healthy. So all this diversity, what's the consequences of all this diversity that we have discovered over the last 15 years? Most of these mutations that here you can see in green have no known phenotypic effects. Each of us, regardless of our ethnic or geographic origin, will differ from each other at around 3 million differences. So what about differences between individuals? If one takes two individuals at random, regardless of their ethnic or geographic origin, there will be 3 million differences between every two pairs of individuals. Most of these differences have no or not known phenotypic effects. So the study of these so-called neutral mutations between populations is very useful to understand human origins and migration patterns. But some of these mutations that you can see here in red will have a phenotypic effect, either because they change the protein or the levels of gene expression, and they can be involved in phenotypic differences such as benign phenotypic differences, such as simply our different physical appearance, but also some of these mutations can be involved in other phenotypes, such as our different adaptations to climate, the different ways in which we are going to digest some food, like milk, alcohol, the different way in which we are going to respond to therapy, and then we get into the field of pharmacogenetics. And something that we are particularly interested in is how these mutations of the humans can be involved in the different ways in which we are going to deal with infection. So all this diversity that there is between individuals and populations, which are the forces that drive this diversity? We have genomic forces, such as mutation and recombination. We have demographic forces, such as a population that is under demographic expansion will show much more diversity than populations that have gone through bottlenecks or founder effects. And also we have natural selection, that is, some mutations that will help us to better survive and reproduce in a given environment. All these forces, genomic, demographic, and selective forces, do influence the patterns of diversity of the human genome, which in turn can influence phenotypic diversity, both within populations and between different human populations. So what do we know today about genetic diversity and population history? We know, thanks to archaeology and also and mainly genetics, that we all, modern humans, originated in Africa around 200,000 years ago. So here you have a summary of the main migrations of humans when they left Africa to colonize South Asia and Australia very early, Europe, East Asia, to finally reach the Americas. But these migrations were not so easy, because humans had to adapt to different environments, climatic environments. Humans had to adapt to different nutritional resources, and humans also had to adapt to different pathogens they encounter all along their migrations. So first of all, let's define now the different forms that natural selection can take. In this slide, you have chromosomes with mutations, and mutations that are in blue, that are neutral mutations. That means mutations that have no effect on phenotypes. In the upper part of the panel, you have the first form of natural selection, that is purifying selection. You see that there is some mutations that are in black that are deleterious mutations. So as you can see, by purifying selection, there will be a purge of deleterious mutations to get rid of deleterious variants. Then you have other forms of genetic adaptation, such as positive selection or balancing selection. Let's take the example in the middle, positive selection. You can see that you have different chromosomes with neutral mutations in blue. But you can see that there is a mutation in red that appears. And this mutation is advantageous, allows us to better reproduce and survive, let's say because this mutation confers a better resistance against malaria. So as you can see, this mutation will increase in frequency in the population to allow the population to get better resistance against malaria. Let's take an example of positive selection. And for that, let's go back to Africa, 70,000 years ago, where we were all living there. Under neutrality, that means the absence of natural selection, here you have an African population with some blue people and some red people. So the frequency of the red allele, if we make it arrive until today, will fluctuate between Africans on the one hand and Europeans on the other hand by simple genetic drift, which means simply stochasticity. So if you take today the genomes of African individuals and European individuals, you will see that there's almost no differences in the allelic frequencies between most of the mutations of the human genome. Now let's concentrate on the right panel, and let's go back again 70,000 years ago. And we have here a mutation in red, that this mutation is not neutral. This mutation confers a selective advantage. And let's imagine that this mutation confers a selective advantage to better survive malaria. So both Africans and Europeans inherit this mutation. But here there's a difference, and it's the environment. And let's imagine that there is just malaria in Africa and not in Europe. You can put it the other way around if you want. And let's arrive until today. The mutation in Africa will increase in frequency to provide resistance to malaria, whereas in Europe it will remain at low frequencies or even disappear. So in exceptional circumstances, there can be mutations that are extremely differentiated between populations, and this is suggestive of the action of population-specific positive selection. In doing so, we could identify genes that have played a major role in human population differentiation, genes that have kind of obvious phenotypes like morphological traits, a phenotype that we do know that varies between human populations, skin color, hair development, but also we could identify genes that may have a major impact on disease-related issues, such as genes involved in metabolic syndrome, obesity, diabetes, hypertension, but also genes involved in immune response to pathogens. An iconic example of positive selection is related to milk consumption, and more specifically to the way lactase activity persists in adulthood in some populations. The genetic basis of lactase persistence are very well known. In Europe, as well as in several parts of Africa and the Middle East, independent mutations have been targeted by positive selection to allow human populations to drink milk in adulthood. Another nice example of genetic adaptation at a microscale level is adaptation to life at high altitude. An example is Tibetan populations that live at elevations of around 4,000 meters. Oxygen levels are much reduced. There are several mutations, including in the gene IPAS1, that allow Tibetan populations in contrast with Han Chinese populations that live in the lowlands to live at high altitude. And different independent mutations in different genes have been found in populations of Native Americans and also in Ethiopians living at high altitude to allow these populations to live in such extreme conditions. So now let's talk about how natural selection and the study of genetic adaptation can be informative to understand infectious diseases. There are some microbial factors that are involved in the outcome following infection of the host. Of course, exposure factors. We need to be exposed to a microbe to develop an infection, but also every microbe can have different virulence factors. But we are particularly interested in the host genetic site of the susceptibility to infection. From the host side, we can have non-genetic factors such as age or sex that can influence the outcome of infection, but we do have also genetic factors. And this is one of the main interests of my laboratory here at the Pasteur Institute. Why do we care about selection and infection? Because infection has been a major cause of mortality all over human evolution, until basically yesterday from an evolutionary point of view. For example, in the UK, in the end of the 19th century, only 38% of the population reached the age of 40, whereas today, 98% of the population reached the age of 40, thanks to the technological advances in the beginning of the 20th century, including vaccination, hygiene, and antibiotics. This does not mean that infectious diseases are not anymore a major cause of mortality, because as you can see here, in the same year as the UK, Mozambique, in the 2000, just 50% of the population reached the age of 40. There's classical examples of natural selection related to infectious disease. One is the heterozygote advantage of the hemoglobin S allele, that in the heterozygote state, that is, S allele and non-S, people are higher protected against Plasmodium falciparum malaria. But there's a price to pay for that, because people that are homozygous for the HBS allele will develop sickle cell anemia. Over the last years, we have been massively working on innate immunity and how selection has targeted the different innate immunity players. Why innate immunity? Because innate immunity is the main line of host defense against pathogens. So we'll be dissecting the form and intensity in which natural selection has been targeted the different receptors of innate immunity. And this has allowed us to make a hierarchical rank of genes here in red that play a major role in host survival, with respect to others here in gray that display higher levels of immunological redundancy. By immunological redundancy, we don't mean that they are useless. We mean just that they are less important and they are redundant. When they are not there, or when there is mutations that makes that this gene lose function, other genes will take over. We have also been able to identify particular mutations that have given and confer a specific selective advantage to a given population. Here, for example, we identify a haplotype in 3 genes of the innate immunity system, TLR1, 6, and 10, that provide a specific selective advantage to European populations. By doing some functional analysis, we show that this selective advantage is related to a reduced level of immune response, which raises question about the good trade-off of innate immunity between well-sensing a pathogen, well-recognizing that a pathogen has got into your body, but not exaggerating too much inflammatory responses because they can be harmful for the host. Finally, another question that we are interested in is how Neanderthals have shaped our immune responses. We know today, thanks to the massive amount of work on ancient DNA, that our genomes of Europeans and of Asians can have up to 6% that derives from archaic hominins, such as Neanderthals or Denisovans. But we know very little about how this, what we call introgression, that means the introgression of Neanderthal or Denisovan genomes to the genomes of modern humans, contributed with immune response alleles. One question that we are particularly interested in is how Neanderthal has contributed to modern humans with advantageous immune variation. This work has been possible thanks to the massive amount of work that has been done with ancient DNA in the last 10 years. We know today that the genomes of modern Europeans and modern Asians and Southeast Asians have up to 6% of ancestry from Neanderthal and or Denisovan genomes. But we know that this 2 to 6% of archaic ancestry, it's an average across the genome, and that this average can go some places and some regions of the genome can have 0% of Neanderthal ancestry, whereas other regions of the genome can have up to 64% of archaic ancestry. My laboratory has been particularly interested in understanding how this Neanderthal introgression in our genomes have affected immune responses. In particular, we have shown that innate immunity genes, both in Europeans and in Asians, have a higher amount of Neanderthal ancestry than the remainder of the genome. And this has allowed us to identify some genes that present very high levels of Neanderthal ancestry and that they are involved in major innate immunity pathways such as TLR1, 6, and 10. Today, the standard approach to treat patients is to treat them all with the same treatment. This makes that, for example, upon treatment with different individuals with different genetic backgrounds, as you can see here, you will have, for example, just 20% of the population that will have a positive outcome of the treatment. There can be others that have secondary effects. For others, the treatment is toxic or simply non-effective. So the idea of precision medicine is to stratify the population by different categories, and in order to give them all the same treatment, tailor the treatment in a way that is adapted to their genetic background, in order to improve efficacy and to reduce side effects. But for that, and to reach this dream that will have a major impact on public health policies, we need to first understand precisely the relation between genotypes, that means our genetic background, and phenotypes, that is, in this case, the way we respond to a given treatment. One of the phenotypes that we are particularly interested in the context of precision medicine is immune response variation. There is a huge amount of variability between individuals and populations in the way we respond to infection. Some individuals, as you can see here, can respond too much to an infection or to a given vaccine, and we call them hyper-responsiveness individuals. But some individuals here in the left can respond too little to infection or to treatment, and they are hyper-responsiveness individuals. Understanding that factors, genetic, epigenetic, environmental, that make that some individuals can respond too much or too little to infection, to vaccines, will be a major goal in the next years. To do so, there's different methods to understand genotype and phenotype correlations. There's classical clinical genetic studies whose main aims are to identify variants that are rare, sometimes family clustered, and once they are identified, these variants are usually associated with very severe phenotypes. Typical examples are mutations in interferon gamma or the toll-like receptor 3 that are associated with several life-threatening diseases such as Mendelian susceptibility to mycobacterial infections or herpes simplex encephalitis. Then we have epidemiological genetics approaches, or GWAS, by genome-wide association studies. The goal of GWAS are to identify variants that are common in the population and that increase or decrease susceptibility to common disorders. Today, thanks to GWAS, there's more than 13,000 1,000 SNPs that have been already associated with more than 900 traits or diseases: height, eye color, malaria, tuberculosis, and many diseases involved in metabolic traits such as obesity, diabetes, or hypertension. But we have other approaches that we can use to better understand genotype-phenotype correlations. These approaches are based on the study of people that are healthy, the general population. We can divide it in population genetic studies whose main aims are to understand how natural selection has targeted specific genes or gene families, or cellular genomic studies, whose main goals are to understand, for example, how gene expression will be altered after a given infection, how dendritic cells, for example, are altered if we treat them with Mycobacterium tuberculosis, to ultimately understand how the genetics of these cells, that means the genetics of the human host, will dictate or at least be associated with the variance in gene expression following, for example, Mycobacterium tuberculosis infection. So the future is now to integrate multiple lines of different evidence. We need to understand how lifestyle and environment of different human populations affects the way they are going to respond to infection. We need to understand how their genetic diversity, their epigenetic diversity, their microbiota also influence the ways they are responding to infection. We need to much better understand the degree of variability in immune responses between individuals and different populations in terms of gene expression related to immunity, in terms of heterogeneity in cell populations, even in terms of correlating genetics, epigenetics, and microbiota to ultimate organismal phenotypes, such as susceptibility to infection, once infected, susceptibility to develop a disease, the clinical outcome of the disease, and the responses to treatments or vaccines. All these together will help us to improve the knowledge on how our genetic background can be used to better understand and improve human health. So thank you for your attention. I hope you enjoyed this talk as much as I did.